Hello everyone, welcome back to Chinmoy's YouTube channel. Now today's episode is about the biological classification part 2. So in yesterday's episode we have already discussed about kingdom Monera and kingdom Protista of 5 kingdom classification. Now today I have told you that I'll be discussing about kingdom Fungi and we are going to discuss about viruses, lichens and viroids. So if you have not yet subscribed to my channel, please do subscribe to my channel and don't forget to like and share my videos. And please do write the comment in my comment box so that if you are having any queries, I can get back to you in the next video. So as of now, let us start with today's lesson, biological classification part 2. So what are the learning objectives of today's lesson? So today by the end of this lesson, we'll know that what is fungi. We will know in details about the kingdom fungi, their types. So what are the groups under this kingdom fungi, and we will be knowing about viruses, viroids, prions and lichens. Now kingdom Plantae and Animalia, though they are according to R.H. Whitaker, they are in 5 kingdoms of classification only, but we are not going to take them in this part because they are having different lesson. We are having different lesson for kingdom Plantae as well as kingdom Animalia. So we are going to learn only about kingdom Fungi and this virus, viroids, prions and lichens in this episode. So this kingdom fungi, the study of anything regarding the fungi is called mycology. Okay. So what are fungi? They are mainly heterotrophic organisms showing a great diversity in morphology and habitat. So they are diverse organisms. They are having so many diversifications that fungi, they can live on moist bread, they can live on rotten fruits, they can live in common mushroom also and on toadstools. So we can say that they are heterotrophic organisms and they show great diversity in morphology and habitat. So from moist bread, rotten fruits, common mushroom and on toadstools, they are fungi. Now if you observe certain mustard seeds, we are seeing white spots on mustard seeds. They are also due to parasitic fungus. So it is a type of fungus that is called parasitic fungus. Now there are unicellular fungi also like yeast, which is used in making bread and beer. Now for some type of special beer and bread, they are always using yeast, which is a beneficial fungi and which is unicellular. They are used to make bread and beer. Now next comes penicillium, which are sources of antibiotics. So penicillium is also a fungi which is a source of antibiotic. Now these fungi are mostly cosmopolitan in nature. So what does this term cosmopolitan mean? You all know that this term cosmopolitan means that they occur in air, water, soil and on animals and plants. So they are occurring in air, water, soil and on animals and plants, and they are mostly growing well in warm and humid climate. So is it clear? So these are the main characteristics of kingdom fungi. So let us move on to the next slide. Now you see here I have taken the structure of fungi in two different this pictures. Now first let us see that with the exception of yeasts, which are actually unicellular, fungi are always filamentous. So this is yeast, which is unicellular, but generally the fungi are always filamentous like this. They are filamentous in nature. So how is their body structure? Their bodies are actually long and they are cylinder thread-like structures. These thread-like structures are called hyphae. So this thread-like structures are called hyphae. Now the network of hyphae is known as mycelium. So if there are many networks of hyphae, they are called mycelium together. And some hyphae, they are continuous tubes filled with multinucleated cytoplasm. So they are having multinucleated cytoplasm and they are called choanocytic hyphae. See, you can see these hyphae, they are actually having continuous tube filled with multinucleated. Multinucleated means they are having many nucleus in one cell. So they are having multinucleated cytoplasm and they are called choanocytic hyphae. Is it clear? So others have septate or cross walls in their hyphae. The others are like they are having septate hyphae. Why? They are septate hyphae. These are not at all multinucleated. So each are having one separate nucleus in each cell. So they are septate hyphae. Now the cell walls, what are the cell walls of hyphae? They are made up of, they are composed of mainly chitin and polysaccharides. So Their cell wall is composed of chitin and they are made up of polysaccharides. So is it clear? So if they are having in a continuous tube, they are having multinucleated cytoplasm, they are called choanocytic hyphae. And if they are separately having the nucleus in each and every section, they are called septate hyphae. Is it clear? Now let us move on to the next slide. Now here, when we start talking about the reproduction in fungi, how it is reproducing. Now mainly fungi, they are reproducing by vegetative means, that is by fragmentation, fission and budding. So these are three vegetative means. Now if asexual reproduction is taking, it is taking by spores called conidia or sporangiospores or zoospores. Okay. So asexual reproduction is also taking place by conidia or sporangiospores or zoospores. Now, when it comes to sexual reproduction, how it is happening? It is happening by the oospores, ascospores or basidiospores. Okay, now various spores are produced in distinct structure which are causing the fruiting agents. So, fruiting bodies, okay. So, various spores are produced in distinct structure called the fruiting agents or fruiting bodies. Now, sexual cycle, it involves the following steps. So, what are the following steps? The first step of sexual cycle is plasmogamy. So, what does this term plasmogamy means? That is fusion of protoplasm between two motile or non-motile gametes. So, the protoplasm is fusing here and that is called plasmogamy. That is fusion of protoplasm between two motile and non-motile gametes is happening. Then what is happening? Karyogamy. So, fusion of two nuclei is called karyogamy. Next to which comes the meiosis in zygote, which is resulting in a haploid spore formation. So meiosis in zygote is resulting in haploid spore formation. So let us proceed with the fungus when it is reproducing sexually. What are happening there? Now in the previous slide we have spoken about the asexual reproduction in fungi. That is how they are making the like fragmentation, fission and budding, how they are reproducing. Now this slide we are stressing on the sexual reproduction in fungus. So how it is happening? Now when a fungus reproduces sexually, what is happening? Now two haploid hyphae, what we have done in that diagram, you just remember two haploid hyphae of compatible mating types, they come together and fuse. So these hyphae, they are coming together and fusing. So what is happening in some fungi? The fusion of two haploid cells immediately results in diploid cells. So two haploid cells, that is n number of cells, that n plus n, they immediately result in the formation of 2n. Now however, in some other fungi like Ascomycetes and Basidiomycetes, what is happening? An intervening sequence or dikaryotic phase or stage is there, that is n plus n, that is 2 nuclei per cell occurs. So such a condition is called dikaryon. So this is a new term, this is a new topic which I am explaining. Please understand it properly, because this is an intervening dikaryotic stage where n plus n or 2 nuclei per cell it occurs. Such a condition is called dikaryon and the phase is called dikaryophase of the fungus. Okay, so this phase is called the dikaryophase of the fungus. Now later what is happening when this dikaryon is formed and the dikaryophase of the fungus is formed? Later the parental nuclei fuses and the cell becomes diploid. So the parental nuclei is also fusing with this nuclei, two nuclei, and it becomes diploid. Now fungi forming the fruiting bodies in which the reduction division occurs, leading to the formation of haploid spores. Now the fungi is forming the fruiting bodies and which in reductional division which occurs leading to the formation of haploid spores. So now we are coming to the different types of fungi which we have discussed here. So here till now we have discussed about the morphology, we have discussed about the structure, we have discussed about the sexual as well as the asexual reproduction in fungus. Now comes the individual categories of fungus. Now the first category comes the Phycomycetes. So what are these Phycomycetes? These Phycomycetes, they are found mainly in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. Okay, they are always found in the aquatic habitats on decaying woods and in obligate parasites on plants. Now, mycelium. What is mycelium? Mycelium, it's an aseptate. So, aseptate means it is having no septum and coenocytic. So, coenocytic, what does this term mean? That is the multinucleate cell that can result from the multiple nuclear division. So, we have seen just now that a coenocytic hyphae, it was there. So, what is coenocytic? That is many number of nucleus, that is multinucleate cells that can result from multiple nuclear divisions. Now, asexual reproduction, it takes place by the zoospores and they are motile, and by aplanospores which are non-motile. So, asexual reproduction is taking place in this Phycomycetes by zoospores, that is motile, or by aplanospores, that is non-motile. Now, spores are endogenously produced in a sporangium. Spores are endogenously always produced in sporangium. A zygospore is what? Is a fusion of two gametes. A zygospore is a fusion of two gametes. Now, and the similar in morphology is called isogamous, and which are dissimilar in morphology are called anisogamous. So, Phycomycetes which are similar in morphology are called isogamous, and which are dissimilar in morphology are called anisogamous. Now, what are the main examples of Phycomycetes? They are Rhizopus, Albugo, and what is Rhizopus? It is mainly bread mold. And Albugo, it is a parasitic fungi found on the mustard leaves. So, I have taken the diagrams of these three examples. Now, let us see the diagrams. Now, Rhizopus, it is a bread mold. It is a bread. It is found on the bread mold. So, this is Rhizopus. And this is the sporangium of Rhizopus. This is Albugo, which is found on the leaves of mustard. This is a type of thing which is a parasitic infection on the mustard leaves, and mucus. So, this is also a common example of Phycomycetes. So, let us move on to the next one. Now, next category, what is it? It is Ascomycetes. Now, this Ascomycetes, it is commonly known as a sac fungi, and which are mostly multicellular, and we can say that penicillium, or a rarely unicellular, is Saccharomyces. So, these are under the category of Ascomycetes. They are saprophytic, they are decomposers, parasitic, and coprophilous. So, what is coprophilous? Those which are growing on the dung. So, dung of any animal, they are called coprophilous. So, they are saprophytic, they are parasitic, and as well as they are coprophilous, and they are growing on the dung. Now, commonly these are called sac fungi, and which are mostly multicellular, penicillium, or rarely unicellular. So, yeast, there is another example that is unicellular, is yeast or Saccharomyces. They are saprophytic, and we can say that mycelium is actually branched and it is septate. Now, sexual spores which are formed, they are called ascospores. If you see this diagram, these spores are called ascospores, and asexual spores are called conidia. Now, here you can see this coprophilous growth of this Ascomycetes. This is growing on the dung. So, this is the coprophilous growth of this Ascomycetes. Next to which comes the Basidiomycetes. So, first one was Phycomycetes, then was Ascomycetes, and the third one is the Basidiomycetes. So, this commonly forms the mushrooms and the bracket fungi, or the puffballs. Now, where are they growing? So, the place of growth is mainly the soil, on logs, and on tree stumps. In living plant bodies also, as parasites, we can see like rusts and smuts. These fungi are also growing on this, on the living plant bodies. Now, there we have seen that mycelium, it was also septate there. Here it is branched as well as septate. Now, next comes the asexual spores, which are generally not found, but vegetative reproduction by fragmentation is common. So, asexual spores are here not generally found, but vegetative reproduction by fragmentation is always common. Now, in this Basidiomycetes, what is there? The sex organs are absent absolutely, but plasmogamy is brought about by fusion of two vegetative and somatic cells of different strain or genotypes. It is brought about by the fusion of two vegetative or somatic cells of different strain or of different genotypes. Now, the resultant structure is dikaryotic, which ultimately gives rise to the basidium. Okay. So, the dikaryotic structure of the resultant, which is ultimately giving rise to basidium. So, there are three main examples of this Basidiomycetes. What are they? First one is Agaricus, second one is Ustilago, and third one is the rust fungus or Puccinia. Okay, so let us move on to the example where I have taken the pictures of these three. Now first one, you can see that agaricus is the first one, that is the mushroom. Ostilago, we can see here this ostilago, it is a fungus infection which is formed, and Puccinia, which is also the rust fungus. It looks like actually rust on the leaves of plant. This is called the rust fungus. Now next category, we are moving on to Deuteromycetes. Deuteromycetes, so this is the last type of fungi we have discussed till now. So why is it called imperfect fungi? Because it is having only asexual vegetative phases of this fungi are known. So till now we have seen that all the types of fungi, they are having both asexual parts and sexual parts. But in this imperfect fungi we have seen only the asexual or the vegetative phases are there. Now sexual forms of these fungi were moved into classes they rightly belong to. So they have been moved into other classes which they rightly belong to. And Deuteromycetes, they reproduce only by, we have told that only asexual spores known as conidia is there. In the previous slides also, if you try to recall, there are conidia, there are mycelium, and there are sexual forms also. But here only the asexual spores known as conidia is present. The mycelium is septate as well as branched. The previous one also, the mycelium was septate and branched. Now some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. So major part, they are saprophytes or they are parasites. Okay, either they are parasites or they are saprophytes, and they are large number, they help into litter and help in the mineral cycling. Okay, and what are the main examples? The first main example is Alternaria, Coletotrichum, and the Trichoderma. The three main examples are Alternaria, Coletotrichum, and Trichoderma. So now let us move on to the next part where we have taken the diagrams of these three examples. Now see here, Alternaria, it is having this type of spot, this type of fungus is having a spot on the green leaves. Then comes the Coletotrichum. These are all strains of Coletotrichum, and this is Trichoderma. So these are the three examples of Deuteromycetes. Now we will move on with the two other main things, that is the viruses, virions, and the lichens. Now in this 5 kingdom classification, till now what we have classified, that's kingdom Monera, Protista, Fungi, Plantae, and Animalia, there have been no mention of these viruses, viroids, or lichens. So why there was no mention of these things? Because some are acellular, and like others, virus, viroids, and prions are made. So viruses didn't find any place in classification, as they are not considered as a true living thing. So only in classification we have taken the living things. The viruses are non-cellular organisms that are actually characterized by having an inert crystalline structure outside the living cell. So they are having an inert crystalline structure outside the living cell. Once they are infecting the cell, what is happening? They are taking over the machinery of the host cell to replicate themselves by killing the host. Okay, so they are infecting the cell. What they are doing, like viruses, they are infecting the cell. They are taking over the machinery of the host cell to replicate. That is, they are dividing, they are replicating themselves, and they are killing the actual host cell on which they are replicating. So what does this term virus means? The viruses means actually venom or poisonous fluid. Now this scientist Ivanovsky, in 1892, he recognized that certain microbes as casual organisms of mosaic disease of tobacco are there. Now these are actually smaller than bacteria, and as they have passed to the bacteria proof filters. So they are passing through the bacteria proof filters, and they are smaller than bacteria. The extract of the infected plants of tobacco could cause infection in healthy plants, and they are named as a new pathogen virus and are called the fluid as Contagium vivum fluidum, that is infectious living fluid. So what the extract is called? These infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus, and they are called the fluid as the Contagium vivum fluidum, that is the infectious living fluid. So it is clear to everyone that what are viruses and how they are causing infection to the living world. Now we are moving on to the next one. Now this W.M. Stanley, what he did in 1935, he showed that viruses could be crystallized, and crystals, they consists of large proteins. Now viruses, they are inert outside a specific host cell, so they stay inert outside their specific host cell. Once they attack the host cell, they are trying to kill the host cell with their venomous liquid. Now they are obligate parasite in nature, and in addition to proteins, we can say that viruses also contain genetic material that could be either RNA or DNA. Okay, is it clear? So in addition to proteins, viruses also contain a genetic material that could be either RNA or DNA. And no viruses contain either RNA or DNA. So there are no viruses that they are not containing RNA or DNA. So they could either have RNA or DNA. And a virus is a nucleoprotein, and it is having a genetic material which is infectious in nature. Now viruses that infect the plants have a single stranded RNA, while viruses that infect animals, they have single or double stranded RNA or double stranded DNA. So the viruses which are infecting plants, they will always have a single stranded RNA, whereas viruses that infect animals have either single or double stranded RNA or double stranded DNA. So now we'll move on with the other parts. Now bacteriophage, or we can say that the phage virus, what are these? These are actually the bacterial viruses or bacteriophages, which are the viruses that infect the bacteria, and they are usually double-stranded or the double-stranded DNA viruses. Now they are having a protein coat. Now you can see here, when you are seeing this label diagram, you can see they are having a protein coat, and this is called the capsid head, which is made up of smaller subunits called the capsomeres, which protects the nucleic acid. Now these capsomeres are arranged in helical and polyhedral geometric forms. So this capsid head, these are all arranged in polyhedral or helical geometrical shapes or forms. Now mumps, smallpox virus, herpes, influenza, AIDS, these all are the different kinds of virus which are having pathogenic effects on human beings. Now in plants like this only, we are having mosaic formation in the leaf, okay, rolling or curling of the leaves, yellowing of the vein, clearing or the dwarfing or stunted growth of the plant. So these all are the effects of several viruses on plants. So as in animals, we are having several viral infections like mumps, measles, pox, influenza, AIDS and all. And recently we have also seen the severe effect of COVID virus, so COVID-19 virus. So like that only, plants are also having virus infection, which are causing the curling and the yellowing of the leaves, veins clearing, dwarfing or stunted growth. Now after viruses comes the viroids. So what are these? In 1971, T.O. Diner discovered a new type of infectious agent that was much smaller than virus, and that caused a potato spindle tuber disease. So this was invented by Diner, the scientist. It was found that they are having free RNA, but it lacked the protein. So as in the previous one, in bacteriophage, that virus, we have seen that they are having protein which are forming the capsids, and they are having a helical or polyhedral shape. But in this viroids, they are not having any RNA, the free RNA they are lacking the protein coat that is found in viruses. Hence the name is viroid. And the RNA of the viroid was of very low molecular weight. So the RNA, which was a free RNA which was found in this viroids, it was having a very low molecular weight. Now next comes prions. What are these? Now in modern medicines, this is a very important name. In modern medicine, certain infectious neurological diseases have been found which are transmitted by an agent consisting of an abnormally folded protein. Now if you see this diagram, you can see that this protein is abnormally folded. So this abnormally folded protein, this was helping to transmit a serious type of infectious neurological disorder. So from that only, the agent was also similar in size to the viruses, and they were called prions. Now this disease caused by prions is called the BSE, or bovine spongiform encephalopathy. So this kind of neural disease or brain disease which was occurring, it was called bovine spongiform encephalopathy. So please learn this term if it comes in your exam, so that you can write what is BSE disease. So it is caused by prions, which are the agent which are having abnormally folded protein, and this is called bovine spongiform encephalopathy, which is commonly called the mad cow disease in cattle and is an analogous variant of this Jacob's disease in humans. Okay, so this is all about prions. Now let us move on to the last part. So here we are in the last part of this lesson, that is lichens. So lichens live in symbiotic association, and they stay in useful association between the algae and the fungi. And the algal component is known as the phycobiont and the fungal component as the mycobiont, which are autotrophic and heterotrophic respectively. Now this algae prepare the food for fungi, and the fungi provide shelter and absorb nutrients and water for their partners. So they are in close association between the algae and fungi. So this type of fungi, what they are doing, they are in staying with close association with the algae, and algae is preparing the food, whereas the fungi is absorbing the nutrients and water for its partner. Now lichens are very good as pollution indicators, because they do not grow in polluted plants or polluted areas. So they are very good pollution indicators. Where there is pollution or where there are polluted plants, the lichens will not grow in that. And the lichens are always staying in symbiotic association. So this is all for today's lesson of this kingdom fungi, and all about viruses, viroids, prions and lichens. So please go through it. Don't forget to subscribe to my channel, so that the next lesson, when I am coming with kingdom plantae and animalia, it will be easier for you to do all the lessons in a progressive manner. So stay tuned, stay updated with my lessons, and don't forget to subscribe to my channel. Thank you.